Welcome to this early researcher seminar series hosted by Cambridge Reproduction. We are very grateful to all the volunteers that are organizing and coordinating this series, and to all the speakers that have agreed already to take part. The seminars are generally held online every third Thursday of each month at 1 p.m. And there are still the details of all the forthcoming seminars can be found online, and I believe that the link will be soon also posted in the chat, link to our website. And just a few notes about what Cambridge Reproduction is. Cambridge Reproduction is an interdisciplinary initiative that brings together researchers from across the university with an interest in any aspect of reproduction, whether it's science, technologies, but also medicines to arts and humanities. The membership is open to all staff and postgraduate students, and you've got access to event funding schemes and regular newsletters. And you are very welcome to join. And again, you can find further details on the membership on our website. And there's always a lot of interest in receiving new abstracts for future seminars. So if any postgraduate student, postdocs would like to take part in a seminar, please also just follow the link that will be posted in the chat and be in touch with us. So our second speaker is Timo Kohler on cells in gels, identifying novel regulators of early embryonic development. Timo trained as a biochemist during his undergrad and master studies, and subsequently is specialized in stem cell biology and early embryonic development during his PhD at the University of Cambridge. His work is focused on how pluripotency is established, maintained, and executed. So over to you, Timo, and thank you for being with us today.
Yeah, so I will be talking about cells and gels and how we use those cells and gels to identify novel regulators of early embryonic development. I'm currently doing a postdoc in Florian Höfler's lab at the Department of Biochemistry here at the University of Cambridge. And to give you an idea of the few next steps we're going to be talking through is there are three parts to this little talk presentation. On the one side is we're talking about why do we study pluripotency? What is it? Where's it coming from? And why is it important? How we're using droplet microfluidics to investigate pluripotency, and what we learned from using droplet microfluidics to about cell adhesions during early embryonic development, and how it actually also influences pluripotency during early embryonic development. So from my understanding, it's always a quite heterogeneous audience in this case. So I will give a bit of background about where does pluripotency come from and where does it originate. So if we're looking at a mouse embryo in the early days, this is that projection. So you see all the cells of the embryo. You can see there's a bunch of cells basically in a sphere aggregating. If we now take a single Z slice, so we just take one layer of this embryo and have a look, you can see there's a big cavity in the center and these cells on the outside called the transcriptome, which go on and form the placenta, plus then a bunch of cells that are clustering here right at the top. So if we look at closer to these cells that are called the inner cell mass, they actually further segregate into two different cell layers. On the one side, we have the primitive endoderm, which goes on to form the hypoblast, the yolk sac. And the other cells right in the center, the precious purple ones, are called the epiblast. And so these epiblast cells are the in vivo origin of pluripotency in the embryo. And they are the cells that then go on and form the whole organism. Basically, all the cells in your body are coming from these original cells found here in the early epiblast. And despite decades of research in vivo and in vitro, we still not fully understand where pluripotency, or how pluripotency is established and then maintained and also executed. Luckily, because embryos are hard to come by, especially if we're moving away from the mouse and looking into human. For example, we can also work with embryonic stem cells. So in the beginning, we're going to be talking about mouse embryonic stem cells. These are derived from the pre-implantation embryo, about this epiblast cells that we were just talking about. You can keep them in vitro, basically indefinitely in culture, maintaining this pluripotent potential, meaning they can also in vitro differentiate to all germ layers, and also actually the PGCs that Valentina was just talking about. And again, thanks to Valentina, we don't need to talk that much about droplet microfluidics, but in our lab, instead of using conventional tissue culture plastic, we're actually using droplet microfluidics to investigate pluripotency. And in this case, what you can see on the left is, I mean, cells grown normally are on two-dimensional tissue culture plastic, which really does not resemble what is going on in vivo in most scenarios, and certainly also not what is going on in the early embryo. So instead of culturing them always on tissue culture plastic, we actually use microfluidic devices to encapsulate single or multiple cells into spherical hydrogels. And the principle is exactly the same from what Valentina just said. Instead, so that instead of having just a liquid where we then use it for sequencing, we actually use a hydrogel precursor together with our cells. And upon, in this case, cooling, these form spherical little hydrogels that can then contain single or multiple cells in 3D. I don't know how many cell biologists or embryologists are with us at the moment, but normally many people don't really understand how the setup is and think it's quite complicated. But to be honest, it's very straightforward and easy to use, and almost everybody can quite easily set it up. So what you can see here is a sterile workbench. We have a regular light microscope, a camera to actually see what's going on. Here you can see a double inlet chip, because in this case we use 2 different cell types, and precision pumps to basically control what goes in and out of these channels. If you then look at one of those microflake chips, this is what they are. You can see one single microscopy slide. It's quite small. We make them in-house. PDMS, it feels rubber-like, but it's actually from the structure very similar to glass. That's why it's nicely bonds to a glass cover slip. And then the tubing that we use to control the flow on the channel. And we've had several projects over the last couple of years working with this microfluidic encapsulation technology. Together with Lucia, we work actually on mouse liver organoids. We have also had a project where we're working on human pluripotent stem cells. And currently, together with Clara and Erin, we're in the process of writing up a story about amnion formation and early embryonic development in marmoset. And one of the great things about droplet microfluidics, it's a high throughput system. So we can single cell encapsulate or multiple cells per microgel in a high throughput fashion. So normally we make between 150 to 300 gels per second, which gives us a good amount of material to work with eventually. In this case, we are having completely chemically and also physically defined 3-dimensional matrix. We have compatibility with other microfluidic systems that are in development or also already produced. And we have the spatial confinement where we can take a single cell or multiple cells and have them confined in a matrix in this case. And if this is still not convincing enough, actually, there was a master student in our lab who then went on and used this technology and commercialized it, Aurora Biotechnologies, which is a very much a plug-and-play system, where basically on one a lot more professional-looking chip than the ones we make in-house, you can do the whole procedure of encapsulating cells and analyzing and imaging afterwards. So back to the original question is, how is pluripotency established, maintained, and executed? And in this particular case, we're caring about, does the 3D spatial confinement influence pluripotency in the beginning in embryonic stem cells? So to do so, we encapsulated mouse embryonic stem cells that were normally grown on plastic in 2 different culture conditions, 2-alif, which is a medium that contains cells in a naive pluripotent state where they're all homogeneously in this very pluripotent state, and also cells in serum lift, where there's a lot more heterogeneity. That's why we call it metastable pluripotency, because some of them are pluripotent, where the others are on the path towards differentiation. And what you can probably appreciate by just going into the microgel system, the morphology changes quite dramatically and almost looks a little bit more of what we have seen here in 2-alif, whether it's on plastic or the gel. What we then realized is, by going into the microgel system, not changing anything else, we actually have an increase in, we have in this case a REX1 reporter line. REX1 is here in this case our readout for pluripotency, because it's one of those genes that comes on during the pluripotent state in the embryo. Just by going into the microgel system, we actually have an upregulation of this protein in the cells. Yes, this is just one protein. We do not know if this is actually a case for everything else. So we also looked at a bunch of others, in this case, KLF4, which is another naive pluripotency marker, just like REX1. And you can see by going from plastic tissue culture and 2D into these microgel systems to again have an upregulation of another naive pluripotency marker. And we start to wonder, is this now like a general trend? And also we had, of course, no idea what is actually causing this boost of pluripotency in this case by just going into a three-dimensional confined microenvironment. So we did also a bit of sequencing. This is not like Valentina single cell sequencing, this is actually bulk sequencing. And don't be overwhelmed by all these names, it doesn't really matter. All it does need to show you is that between plastic and 3D, there's a bunch of differentially regulated genes, again, by just changing the way we culture the cells instead of, I don't know, differentiating them or changing the medium. We really just put them into the microgel system. And you can see things like ESR beta and KLF2, and I think there was another one there, the KLF4 that we talked about. All these are naive pluripotency genes. But one of the genes that we quite quickly saw and identified is a gene called JUP. So JUP encodes a protein called Plakoglobin. And most of you probably never heard about it. And I have to thank Joe, who's our sequencing guru, who initially pointed this out. Because, yeah, in the beginning, you could, of course, say, oh, which one I'm going to look at it, because any of them is probably good as any other. However, Plakoglobin is actually a vertebrate homologue of beta-catenin, and most cell biologists or embryologists have definitely heard of beta-catenin. And you will appreciate they are quite similar. I mean, there's 70% sequence identity match between beta-catenin and Plakoglobin, both evolutionary conserved. But Plakoglobin can sit just like beta-catenin in Heron's junctions, but also in desmosomes. If you have mutations in Plakoglobin, you get diseases like ARVC, which is arrhythmogenic right ventricular cardiomyopathy, which is a heart disease. Depending on the context, Plakoglobin can act as an oncogene or as a tumor suppressor, which is surprising because beta-catenin is a very well-studied oncogene. We know that Plakoglobin has mechanoresponsive capabilities in xenopus, and both proteins are actually embryonically lethal if they are knocked out, just at very different stages. So we thought maybe it's a good idea to start looking into Plakoglobin a little bit closer. And this is what you get if you type in beta-catenin or Plakoglobin into PubMed. And you will appreciate on the left, beta-catenin keeps growing and everybody very much cares about it, whereas its sibling protein Plakoglobin has a lot less interest, I would say. And if we actually adjust the axis to match beta-catenin, this is what we get. I mean, for me, this could be either a reason for concern or a reason for excitement. I decided to go with the latter. And indeed, if we culture mouse embryonic stem cells in these microgels, we actually do get a phenotype. It's not just at on sequence level that the transcript seems to be coming up, but also the protein level seems to be upregulated, whereas vice versa, beta-catenin seems to be slightly downregulated, although we didn't see a change in transcription level. So the question is, can Plakoglobin actually regulate pluripotency? And for that, I'm not going to go into the nerdy nitty-gritty detail too much about embryonic stem cells. You have to believe me a little bit on this one. We made an overexpression construct for Plakoglobin. We chucked it into our embryonic stem cells. And just the pure overexpression of Plakoglobin was enough to reestablish this homogeneous state of naive pluripotency, even under serum lift conditions. So you can see here, these are the wild type cells, where some of the cells express KLF4, just like we had seen before. If we overexpress Plakoglobin, suddenly all of the cells start expressing again this naive marker, KLF4. Because we are on a reproduction talk right now, I'm going to focus a little bit more on the embryo for the rest of the talk. And meaning, does Plakoglobin actually play a role during early embryonic development as well? So for that, we started looking into embryonic development. In this case, we're going to be looking at the mouse. And what you can see here is different stages of pre-implantation development. This is day E2.5, which is 2.5 after fertilization. And you can see there is no Plakoglobin expression at all. As soon as we reach this early blastocyst stage, we have upregulation of SOX2 and SOX17, which are epiblast and primitive endoderm markers respectively. And in green, you can see also upregulation of Plakoglobin. Now, the Plakoglobin that we see here on the outside was not too surprising because, as I said before, Plakoglobin is also found in desmosomes, and the trophectoderm is very much enriched in desmosomes because it needs to hold the structure of the embryo. However, the inner cell mass here does not have desmosomes. So the question was, okay, plakoglobin suddenly also seems to be appearing here in the epiblast, the inner cell mass cells, the origin of naive pluripotency also in vivo. In comparison, beta-catenin is expressed from very early stages on all the way to expanded blastocyst stage at E4.5 in basically all cells. Because we were wondering, and we had seen this regulation of plakoglobin and connecting it to pluripotency, we were then wondering, if it starts to be expressed during this naive stage of pluripotency in the pre-implantation embryo, what happens as soon as the embryo actually implants into the uterus and transitions out from naive pluripotency into prime pluripotency? So for that, we had a look at post-implantation, in this case, still mouse embryos. And what you can see is, right here where the line is, it's a cutoff between the epiblast, those are the cells, again, that go on and form the embryo, and then extra-embryonic ectoderm, which is extra-embryonic tissue, so it doesn't go on. Suddenly, the epiblast, which was 24 hours before highly enriched in plakoglobin, suddenly has lost it completely. It's just disappeared. Whereas it also does not come back at later stages. I mean, the next time it will properly show up again is during heart development. In contrast, beta-catenin stays expressed throughout post-implantation development as well. So we do not see this difference where plakoglobin suddenly disappears during the transition from naive to prime, but rather it just stays on constantly. So we're wondering, can we actually see the same also in vitro? I had said before, we used embryonic stem cells as a model for pre-implantation naive pluripotency. Vice versa, you can use prime pluripotent stem cells called epiblast-derived stem cells by deriving them from this post-implantation embryo. They have different signaling requirements, but very nicely match of what's going on in the post-implantation embryo. And indeed, just like we'd seen in vivo, these cells do express beta-catenin, E-catenin, which is a transmembrane protein where it's normally located, but have completely, again, lost plakoglobin on protein level. So wondering, we had a bit of meta-analysis from another paper, Stuart et al. 2019, where they analyzed these cells on single-cell sequencing level. Surprisingly, plakoglobin does get downregulated by the transition from naive to prime cells. However, it's never as striking as you actually see on protein level. So we're by now quite convinced that all this regulation, this abrupt regulation, is actually happening on protein level and that the transcripts only catch up afterwards. So we now know that there is a loss of plakoglobin during this naive to prime transition. And myself, personally, I'm quite fond of evolution. And the great thing is about pre-implantation development, it's fairly conserved across the species. So if I show you these 3 different pre-implantation embryos, unless you have very school eye, it's very hard for you probably to tell what embryo actually leads to what organism. In this case, we have a rhino, a human, and a mouse embryo, but in general, they all do look somewhat similar. Please ignore the scales. Of course, neither the schematics nor the embryos are exactly those sizes. If we then go on, we can ask the question, are these plakoglobin expression patterns that we had seen of naive and primed stage in mouse embryonic stem cells and epiblast stem cells in mouse pre-implantation and post-implantation embryos, are they actually evolutionary conserved? To answer the question, together with Jenny Nichols' lab, we then looked in human embryos. And just like in the mouse, in these very early cleavage stage embryos, before the blastocyst expands, there is no plakoglobin. However, beta-catenin expression, just like in the mouse, is already to be found. If we then go on to the blastocyst stage, it becomes quite strikingly that just again, like in the mouse, plakoglobin expression seems to be upregulated. No surprise here out in the trophectoderm, we know it's supposed to be there, but the surprising part was that it is also upregulated in the epiblast of a human embryo. Now, of course, it's basically impossible to look into human post-implantation embryos. But we were still wondering, okay, if we have seen this evolutionary conservation of plakoglobin expression in the naive state, can we also somehow look into that in the primate? So for that, together with Thorsten Borowiak's lab here in PDN, who is a primate embryologist, we then looked into marmoset post-implantation embryos. And as you can see, this is the morphology is quite different from the mouse, but we don't have the time to go really into detail right now. It's actually more like what we see in human. This is the embryonic disc, and the embryonic disc still expresses a pluripotency marker SOX2, but has, just like again in the mouse, completely lost all plakoglobin expression. Whereas in contrast, beta-catenin, its brother, is expression is being basically maintained during the post-implantation stages as well. And we know, just like in the mouse, there is a loss of Plakoglobin upon implantation also in primates. At this stage, we had post-implantation marmoset, no pre-implantation marmoset, and we had pre-implantation human, but no post-implantation human. So to give us a little bit of extra confidence, and we now know that we can actually model these two different stages of pre and post-implantation development also in embryonic stem cells. And to do so, we used naive and primed marmoset embryonic stem cells that were established by Aaron Slattery from Thorsten Borowiak's lab, and human naive and primed embryonic stem cells coming from Jennifer Nichols' labs. And just like we'd seen before in mouse embryonic stem cells and epidermal stem cells, we have the same pattern. However, it's not as striking. So we do still see clear upregulation during these naive state with upregulation of pluripotency. And it seems to be disappearing during the prime states, but there's a bit of more heterogeneity. It's not just blatantly absent like we'd seen in mouse before. Now, one of the things we of course have to consider is that it doesn't matter, even though we call them primed and they represent the post-implantation stages of the embryo development in primates, these cells were still derived from pre-implantation embryos. So maybe it's just a staging thing or timing thing that if we give it a bit more time and keep them in culture, eventually it will also completely disappear. So with that, we can say that Plakoglobin seems to be an evolutionary conserved protein and seems to be always associated with naive state pluripotency. So from what can we basically learn from this project? In our case, we used the microgel encapsulation system as an alternative to conventional tissue culture to ask questions that you wouldn't be able to ask by using these systems. Because, for example, there's spatial confinement that you can achieve in these microgels that you can't achieve on a conventional tissue culture plastic dish. And what we have observed is that encapsulation on this spatial confinement leads to a promotion of naive pluripotency, concomitantly with an upregulation of Plakoglobin. We can then use this newly identified role of Plakoglobin and overexpress in embryonic stem cells and actually re-establish naive state pluripotency. So if we then take this knowledge and went to look basically what's going on in vivo, we realized, yes, even in vivo, in the embryo, we have this upregulation of Plakoglobin in the naive state, pluripotent state, and a complete absence upon transition into post-implantation stages. And this seems to be conserved between at least human, mouse, and marmoset. But for the future, I would be interested, is there also a mechanoregulation in vivo? So what we have seen with the spatial confinement and that needs to be happening to upregulation of Plakoglobin in embryonic stem cells, is there also a mechanoregulation component in vivo? Does it regulate pluripotency in vivo? We do not know at this stage. What are the actual protein dynamics? So we have seen that protein can get quite quickly in the mouse within 24 hours completely disappear. So what is going on there? And then my favorite, how much more of cross-species conservation is there? If we look also into other species, how does the cell-cell adhesion proteins behave? How do they regulate pluripotency? And with that, I would like to thank a bunch of people, in particular Professor Lauren Holfelder, who has hosted me for many years now and always supported our endeavors into cell biology and embryonic development. Of course, Thorsten Borowiak's lab for all the primate work, and Jenny Nickel and Kevin Chalut from the STEMS Institute, and particularly also Joe and Anna, who are my co-first authors on this paper, for all the help. Otherwise, this project wouldn't have been possible. Aaron for all the marmoset embryonic stem cells, and in particular also, I don't know where I have listed her, Ayaka. She has moved now to the University of Tokyo, where she has her own group, because without her help, I would have definitely not learned all the embryo work. With that, thank you very much. And yes, of course, if you have any questions, please feel free to ask now or reach out. And if you want to have a more in-depth look, also have a look at our preprint.